Hi, everybody. I want to do a quick 2-minute overview of the entire cellular respiration process, despite its massive complexity. So if we look at the board, the crazy board here, let us just remind ourselves that what we're focusing on to start are the blue molecules here, which are the ATP synthase proteins on the inner membrane. So again, those are the ATP synthases. And if you remember their method of action, a proton flow causes a spinning action, which leads to a set of shape changes that leads to ATP synthesis. So what needs to be present for ATP synthesis to work, for the ATP synthase, is a high concentration of protons in this intermembrane space. And the way we're going to generate that high concentration of protons is by using this electron transport chain. When fed electrons, the proteins of the electron transport chain undergo a series of shape changes. Again, they go from a shape in the absence of an electron to one in the presence of electron. Absence of an electron, presence of electron pair. Absence of the electron pair, presence of electron pair. And as these proteins of the electron transport chain undergo the shape change, they pump protons into the intermembrane space. That's this process right here. Where the electrons come from and where they go is the last thing in this summary. The electrons are harvested from various fuel sources, whether fats or amino acids or most commonly glucose. As we oxidize, there's the key word, as we oxidize / break down our fuels, we harvest the electrons as we go from 6-carbon glucose to a 3-carbon pyruvate to a 2-carbon acetyl-CoA to single-carbon CO2s. Every step of the way, electrons are harvested and given off to an electron carrier. That's our NADH. And again, we're doing this at all of these steps, at both the glycolysis step, the pyruvate to acetyl-CoA step, and the Krebs cycle step. All of these steps, we're harvesting electrons and giving them to our carrier, which is now NADH. So from front to back, one more time, we oxidize, break down fuels all the way to CO2 and harvest electrons. Those electrons are carried by NADH and its partner FADH2 to the electron transport chain. Diffusion through the matrix takes them to the electron transport chain, where our electron carriers hand the electrons to the electron transport chain. The electron transport chain components undergo shape changes as they pick up and deposit and transfer the electrons, which cause protons to be pumped into this space. The proton gradient going through the synthase in this direction causes the synthase to spin, generating the ATP. The final step of the process, which I just skipped, is where the electrons go at the end of the electron transport chain, which is to oxygen, which is basically released as water. The oxygen is the final acceptor of the electrons in this entire process. Without the oxygen present, the electrons would be stuck here, they'd be stuck here, they'd be stuck here, they'd be stuck on the NADH, and they wouldn't be able to get harvested anymore, putting a halt to all activities inside the mitochondria of your cells. That's the summary. Thanks, everybody.